Welcome. Today, we're diving into a key concept in medicine, the F2 similarity factor. Generic drugs are affordable versions of brand-name drugs, but how do we ensure they work the same way? The answer lies in the F2 factor, a mathematical tool that compares how both drugs dissolve and release their active ingredient. Dissolution is crucial. If a drug releases too quickly or too slowly, it can be unsafe or ineffective. Scientists use the F2 factor to compare the performance of generics to originals, ensuring patients get the same therapeutic benefit. Regulators like the FDA rely on this score to approve generics without expensive clinical trials. In this video, we'll break down what the F2 factor is, how it's calculated, and why it matters. We'll also look at global rules for this test and why understanding F2 is essential for anyone in the pharmaceutical industry. The F2 factor underpins the trust we place in generic medicines, ensuring they're safe and effective for everyone. Let's explore how this simple number protects public health. By the end, you'll see why F2 is a cornerstone of modern drug development. Let's get started. The F2 similarity factor measures how closely a generic drug's dissolution profile matches that of a brand-name drug. Imagine testing both tablets in a beaker that simulates stomach acid, measuring how much drug dissolves at set times. The F2 formula condenses all these data points into a single score. A perfect match would give an F2 of 100, but in practice, regulators set the threshold at 50. If the F2 value is 50 or higher, the two drugs are considered similar in how they dissolve. This allows companies to skip costly human studies and still prove their generic is therapeutically equivalent. The F2 factor is a key part of the biowaiver process, saving time and resources. It gives doctors and patients confidence that generics will work just like the originals. Globally, F2 is a standardized method for ensuring high-quality medicines. It bridges lab tests and real-world drug performance. That's why F2 is so important in generic drug approval. Next, let's see how to calculate it correctly. To use the F2 factor properly, strict rules must be followed. The F2 value must be between 50 and 100 to declare similarity. Below 50 means the generic isn't close enough. At least 12 tablets of both the generic and reference drug are tested to ensure reliable results. Multiple time points are required, usually until at least 85% of the drug has dissolved. If the data is too variable, over 20% at the first time point or 10% at others, the F2 result isn't valid. For very rapidly dissolving drugs, over 85% dissolved in 15 minutes, F2 isn't needed, but you must show the data. These rules ensure the comparison is fair and scientifically sound. Following them is essential for regulatory approval. The F2 factor is only meaningful when calculated under these strict conditions. Now let's see how regulators around the world use F2. Major agencies like the USFDA, EMA, TGA, MHRA, and HPRA all use the F2 factor to assess generic drugs. The USFDA and EMA both require an F2 of at least 50, using 12 units and strict rules for variability and time points. The EMA also emphasizes physiologically relevant test conditions like pH to mimic the human gut. Australia's TGA and the UK's MHRA follow similar standards, making it easier for companies to use one set of data for multiple regions. Ireland's HPRA aligns with EMA guidelines as well. This global consensus on F2 simplifies generic drug development. However, companies must always check for subtle regional differences before submitting products. Harmonized standards mean safer, more accessible medicines worldwide. The F2 factor is a universal language for drug quality. Next, let's see how F2 is used in real-world drug development. For pharmaceutical scientists, the F2 factor is a practical tool, not just a regulatory box to check. During development, formulators use F2 to quickly compare new generics to the original drug. If the F2 value is below 50, they know the formulation needs adjustment, saving time and resources. This rapid feedback loop helps perfect the product before expensive human trials. If F2 remains below 50 despite all efforts, a full bioequivalence study in volunteers is required. In these studies, drug levels in the blood are compared to confirm equivalence. The F2 factor acts as an early screening tool, guiding development decisions. It helps companies focus resources on products that need more testing. Efficient use of F2 accelerates access to safe, effective generics. Now, let's wrap up with best practices and the golden rule of compliance. The F2 similarity factor is a pillar of generic drug development, ensuring generics match their brand name counterparts. Always use at least 12 units, select time points until 85% dissolution, and control variability for valid results. For rapidly dissolving drugs, show the data, even if F2 isn't needed. While global regulators mostly agree on F2 rules, always check the latest regional guidance for subtle differences. Diligent compliance is key to successful submissions. The F2 factor is more than a number. It's a promise of quality and consistency. It enables affordable, effective medicines and builds trust in generics. For students and professionals, mastering F2 is essential in pharmaceutical science. It's a cornerstone of drug quality worldwide. Thanks for watching. Now you know why F2 matters in every pill you take.
If you found this video helpful, please subscribe, like, and share it with others who'd benefit. See you in the next video.